Hello, I'm Ranz from Group 3 for Div MA of Veterinary Parasitology 412. So today I will be discussing the gastrointestinal nematodes of canine.
The materials used in the study are the following: gloves, cover slip, glass slide, test tube rock salt, distilled or top water, plastic cups, graduated cylinder, beaker, weighing scale, fecal sample, compound microscope, and lastly, sieve. So first, what is a flotation technique? Flotation techniques use solutions which have higher specific gravity than the organisms to be floated, so that the organisms rise to the top and the debris sinks to the bottom. The simple test tube flotation technique is a qualitative test for the detection of nematode and cestode eggs, like the Ascarids species and the Trichuris species. Let's begin by collecting a fecal sample weighing precisely 3 grams. Transfer the collected sample into a designated plastic cup, ensuring accuracy in sample size. Second step, to prepare the saline solution, weigh out 400 grams of rock salt. Place the measured rock salt into a beaker, then add 1 liter of distilled water to the beaker containing the rock salt. Vigorously stir the contents of the beaker until the rock salt is fully dissolved in the water. This saline solution serves as the medium for sample processing. After preparing the saline solution, the next step is to enhance sample preparation by adding first a 2.5 ml of tap water to each fecal sample within the plastic cup. Then, utilize a graduated cylinder to accurately measure 47.5 ml of the prepared saline solution. The precise measurement is crucial for consistent and reliable results. Then gently pour the 47.5 ml of saline solution into the plastic cup containing the 3 grams of feces. Using a tongue depressor, thoroughly mix the contents within the cup to ensure even distribution of the saline solution throughout the sample. The purpose of sieving and filtration is to remove solid particles and debris. Perform sieving 3 times on the mixture. This sieving process is integral to obtaining a purified sample. Then after that, fill a test tube with an appropriate amount of flotation solution, ensuring it reaches slightly above the rim of the test tube. The flotation solution is vital for the next steps in the process. Carefully place a cover slip on top of the test tube filled with the flotation solution. Allow the setup to rest for a minimum of 20 minutes. During this time, helminths and other parasitic elements will float to the surface. After the resting period, leave off the cover slip from the test tube. Place the cover slip directly onto a clean glass slide. And after that, proceed to examine the prepared sample using a compound microscope. This microscopic analysis is the key to identifying studying of roundworms and other potential parasites within the fecal sample. By diligently following these comprehensive steps, the flotation technique can be effectively employed for the identification of parasitic worms in fecal samples, serving vital diagnostic and research purposes. This 3-year-old dog has only been dewormed once and lives in a compound with their own kennel. They have a history of mingling with other animals that can freely enter the compound, and their initial deworming was done when they were just 2 months old. We figured out what kind of parasite we're dealing with by comparing it to some nematode class eggs. Turns out, the egg we spotted during our fecal examination belongs to Ancylostoma caninum species. We look at a reference from the Western College of Veterinary Medicine in 2020, and they describe Ancylostoma caninum eggs as having an oval shape with a delicate and smooth shell, which matches our findings. These eggs measure about 55 to 75 micrometers in length and 35 to 45 micrometers in width. Interestingly, Ancylostoma eggs are smaller than those of Eusinaria stenocephala. and this size difference is a handy clue to tell these two parasites apart when inspecting fecal samples. You can actually compare our findings to the ones from the Western College of Veterinary Medicine in the pictures. Ancylostoma caninum, a type of hookworm that dogs can get, is one of the most common parasitic roundworms affecting canines worldwide. When pre-adult and adult worms in the intestine feed on blood, it can lead to severe anemia, and if left untreated, it can even be fatal, particularly in young puppies with heavy infections. Other symptoms include tiredness, loss of appetite, dehydration, vomiting, and poor weight gain. Dogs can get infected with Ancylostoma caninum through various ways, like from their mothers, or also known as vertical transmission, through the skin, or by swallowing third-stage larvae or paratenic hosts. Let's talk about the life cycle of Ancylostoma caninum. So these little sheath larvae get their groove on and become all active in about 2 to 8 days. They thrive in shaded, well-drained soil that's warm and humid. Now these nematodes can infect a host either by being swallowed or by burrowing through the skin. After that, eggs start showing up in the feces about 2 weeks after the larvae get ingested or a month after they penetrate the skin. But not all of them grow up. Some choose to invade muscle cells or the gut wall and be dormant, also known as hypobiosis. These inactive larvae wake up or will be activated later and head over to the small intestine to mature or make a detour to the mammary glands. There, they are shed in the milk and go on to infect the pups. This whole reactivation thing happens frequently during the last 2 weeks of pregnancy. To keep our furry friends safe from these parasites, it's essential to use both environmental prevention measures and give them the right anthelmintic medication. Getting rid of hookworm larvae in your yard or garden can be quite a challenge, but when the weather is mild, you can use sodium borate to get rid of them in gravel or loamy areas. Just keep in mind, it might also harm your plants. There's also this thing called resinated dichlorvos, which can mess with the larva's growth. If you have paved surfaces, cages, or similar spots, a good cleaning and a 1% sodium hypochlorite solution can either kill the larva or make them more vulnerable to the elements. And interestingly, in the world of dog breeding, many commercial operations opt for wire-bottomed cages to keep dogs away from their own waste to prevent them from ingesting the eggs of Ancylostoma caninum. Usually, we protect the environment by regularly treating our pet dogs and cats. We use anthelmintic medication to lower the number of hookworm eggs in their feces, reducing contamination. Dogs can be treated with various products like pyrantel pamoate, fibantel, fenbendazole, melbimycin, oxime, and moxidectin. However, there's a growing worry, as per Jackson in 1987, that in Australia, Ancylostoma caninum, the dog's hookworm, is becoming resistant to parental treatment.